Today we are going to talk about biological databases. A biological database is a database which stores biological information, as the term implies itself. Now, what is the possible biological information that can be stored? As we saw earlier, we have different nucleotides, and every nucleotide will be made up of different bases or different residues. So, the sequence of different bases as well as residues can be stored as some information. It can be of DNA, it can be of RNA. In DNA itself, different types of DNA, it can be chromosomal DNA, it can be plasmid DNA, or in RNA, it can be of tRNA, it can be of 16 srRNA, it can be of small srRNA, or it can be of ribosomal RNA or microRNAs. Like that, any type of nucleotide sequence, the sequence information or what is its sequence, that can be stored in a biological database. Other than the nucleotides, you can have also various proteins, the 3D conformation as well as the amino acid sequence of proteins. What are its structural properties? Let it be a two-dimensional as well as a three-dimensional properties. All these can be stored as a biological information in a database. Other than nucleotide sequences, even at gene levels, you can store the information. Are the genes being found to be homologous in nature? Which are the family of genes which have been present in a chromosome? All those informations come under biological informations. Other than that, you can go to see that when you get a nucleotide sequence, you can say that of course it contains so many nucleotide bases, but at the same time we can also analyze what are the genes there. Further on, you can also say like which processes each gene is being involved, and what are all other informations, what are the promoter sequences, what are the translated sequences here, what are the regulatory elements. All the sequences regarding a gene can also be explained in a biological database. And this process of explaining what are the processes a gene is being involved in it, or what are the other informations regarding that particular gene, refers to a process called annotations. So, when you go to consider a biological database, it can take raw information either of the nucleotide base sequences, information regarding the proteins, let it be a one-dimensional process. Like one-dimensional alignment means the amount of amino acids, the amino acid sequences of a particular protein, the two-dimensional structure of the protein, the three-dimensional structure of the protein. Coming to nucleotides, what are the genes which are being present in it? Are the genes being found to be homologous or heterologous? And in the gene, which process it has been involved? And what are the regulatory elements of those particular genes? All these informations can be stored in a normal biological database. And so you can classify the database in bioinformatics as different databases. They can be considered to be as sequence databases. They can be considered to be as sequence analysis databases, or sometimes you can they can serve as a factor for functional genomics, that is to find out what is the function of every genome. Another type of database which we come across is a literature database, which will store large amount of journals or literature or findings regarding biological informations. It can be structural databases, which shows a database which shows the structural features of certain proteins and all that. It could be metabolic pathway databases, which shares information of various metabolic pathways which have been involved in life, as well as there would be various other specialized databases also, which we'll be talking later on. So, if you go to consider a database is a storage, it's a storage space for lots of information, and since we are storing them with so much data is being stored with using computers, we have to use the term bioinformatics here, because the biological information is being stored in a computer aided assisted database. And the database can be of different types. It can be of a sequence database or a sequence analytic database, as we had mentioned earlier, functional genomic database, or even if you are not taking any sequence or anything other than that, lots of journals. If you are just taking the information of various journals, published journals or published articles on a particular topic and storing it in a database, that can serve as a literature database. Now, basically you can classify the databases mainly into two databases. One is a primary database and the second one is a secondary database. The primary database is usually where the direct submission of experimentally derived data from researchers usually happen. That is, the direct results of all the experiments of, let it be nucleotide sequence, of protein or whatever it is, all that information is directly being stored in a primary database. So, it is also being called as an archival database. You know, archive is something which is related to be old. So, the primary information or the primary raw data which you get after an experimentation, let it be a nucleotide sequence or let it be a protein sequence or different types. So, the primary database or the primary submission of the raw data mainly happens in the primary database. Whereas in a secondary database, it is a result of the analysis, that is a literature research, that is you take the primary data, analyze it, and a literature research and interpretation of various data which is being done in the primary database is being done using a secondary database. And it is also known as a curated database, or it is also referred to as a knowledge database. Now, the examples that we have is the ENA database. It is a European Nucleotide Archives, GenBank, DDBG, Array Express and GEO protein database. These are all examples of the primary databases. Whereas the secondary databases, you have Interpro, Uniprot, Ensemble, etc. What are these? I am not explaining it now. We will be going to the details when we are separately exploring them. So, a primary database is also known as an archival database, and they are populated with experimentally derived data such as the nucleotide sequence, protein sequence or the macromolecular structure. The experimental results are submitted directly into the database by the researchers, and the data are essentially archival in nature. Once given a database accession number, the data in the primary database are never changed and they form of a scientific record. I have told you about the different examples of the primary database we had mentioned earlier. So, every experiment, once a researcher does an experiment and he gets a nucleotide sequence, or let him get a protein sequence, before publishing his results into any journal, he needs to submit all these raw data into a primary database and he needs to get an accession number. Okay, and that such a database is referred to as a primary database. If he use this nucleotide sequence and create further research articles also, he can use the same accession number for the other journals also. So, the primary source of any raw data will be a primary database. Coming to a secondary database, it mainly includes the data which has been derived from the result of analyzing the primary data. Now, the secondary databases, they are often drawn upon the information from numerous sources, including other databases, controlled vocabularies, and as well as the scientific literatures. And they are highly curated, often using a complex combination of computational algorithms, manual analysis, as well as interpretation to derive new knowledge from the public record of the science. Let me just go into the details of a secondary database for you to understand in a better way. You have put lots of DNA sequences in a primary database. Now, consider you analyzed 10 isolates or 10 microbial isolates and you put them into a primary database. Now, if you want to analyze, take all those raw informations and find out what is the what are the relation between them, or what are the genes, or what are the genomic elements of it. You take the data which has been provided in the primary information, and then you do that data, you take that data and analyze it further, and you get some information. That information is mainly stored in the secondary database. Now in the case of you have examples like Interpro, where the protein families as well as the motifs and the domains which have been present in the protein is being analyzed. That is a secondary database. Then you have a UniProt knowledge base here, where the sequence and the functional information of proteins are being put. That is, after the sequence, what are the function of each and every protein is being analyzed using a secondary database here. Then you have ensemble here, where the variation as well as the function and the regulation and more layered onto whole the genome sequence is being done in an ensemble secondary database. Now you should also consider one thing: many data resources may have both primary as well as secondary characteristics. Now for example, in UniProt, it accepts the primary sequences derived from peptide sequencing experiments. However, UniProt also infers the peptide sequences from the genomic information, and it provides a wealth of additional information, some derived from the automated annotation as well as even more from careful manual analysis. So you cannot say like you cannot give a clear cut mark that this is a primary database and this is a secondary database, because sometimes some databases might contain also primary sequences. The example of that was UniProt, which we had exampled. And in addition to this, we also have certain specialized databases. Now for a particular interest, now I'll tell you an example: Flybase, that is one particular database. The HIV sequence database, it is it contains the sequences of HIV alone. Then you have the ribosomal database project, which will analyze the ribosomal data only. So like that, for a special purpose, you have different databases also. So primarily we have different databases, the primary and the secondary. In addition to this, we also have the specialized databases. In the coming sessions, we will be analyzing the different databases and what are the purposes of each and every database using some practical exposure techniques. Thank you for now, students.